Shana Elbaum-Garfinkle is an assistant professor here at the Graduate Center of the City University of New York. She has her laboratory at the Advanced Science Research Center. She received her PhD from Yale. She's a proud CUNY alum, having gotten her bachelor's degree at Hunter in physics. After a postdoctoral period at Princeton, she returned to the City University of New York, to the ASRC and the Graduate Center, where she set up her laboratory in that beautiful building uptown. She has worked on the problems that she'll be talking about this evening for many years now. As with Stephanie, our other speaker, both are in this sort of first generation of young people who have set up labs to explore these new phenomena. Stephanie is an assistant professor at McGill University in Montreal. She received her PhD from Stanford, and she and Shani overlapped as postdoctoral fellows in Princeton, where I got to know both of them. And she too has been working in this field since her postdoctoral years. They haven't actually worked together. They sort of worked in parallel on different parts of the problem. And what you will hear tonight is something of an adventure story and something also of the kind of surprises that science holds for us as we explore more and more. That's a topic which is a very visual and beautiful topic, as you get a hint from the first slide. It's a topic which is of profound significance for our basic understanding of life. And it's a topic which really matters for how we think about human health in the future. So with that, let me ask Shana to start.
Great. All right, thanks so much, Bill. So this is a real treat on many levels, so for one, we all get to take a break from the news cycle. Number two, we get to discuss one of my favorite topics in biology, which is liquid phase separation in biology. It's also a treat because I get to do this with an old postdoctoral friend, currently professional colleague in the field, Steph Weber, and so that's really exciting. And finally, it's exciting as a scientist to do events like these for the public, which are a little more fun, a little less formal, and we can really talk about the science in a different way than we get to normally do. So the goal for this hour is really to expose everyone to this new field, the revolution in cell biology, this concept of cell biology's new phase, which we'll talk a lot about. And I think the hope is that everyone comes away not only with an awareness of this new field, but an appreciation of its awesomeness and its beauty as depicted here. So this is on the right, this mesmerizing hypnotic thing is an artist's depiction of the liquid phases in the cell. But in this case, it's done on a much larger scale, projected onto the dome of the planetarium that exists in the basement of a CCNY building. Just a fun fact. We thought the best way to expose all these aspects of the field to everybody tonight would be to tell some stories, tell some anecdotes about our experiences arriving into a pretty new field. So we both started our postdocs at a point where the field was just emerging. So it's really only a decade old in terms of a field. And so we thought we would do it in sort of three sections, and we'll get as far as we get. So first, we can talk about how we arrived to the field. Then we'll take turns sharing an anecdote of some of the sort of aha moment or a big discovery that we had in the field that really sort of was exciting for us and was important for the field. And end off with just some of what we feel are some of the pressing questions and what our individual labs are focused on now. So before I hand it over to Steph, I'm going to attempt to summarize the revolution in just one slide before we begin, just in case some of you are like, what are we talking about here? And so my one slide, 30-second summary before Steph gets us rolling is that the revolution is actually just a deceptively simple idea, which is that the cells that make up all living things, including you and I, can be thought of or are becoming appreciated to be composed of a variety of immiscible liquids, complex fluids. So something like a really interesting lava lamp or a salad dressing with oil droplets and water, where there's a really growing appreciation that the the liquid that makes up the cytoplasm, the nucleoplasm in the cells are actually these beautiful complex liquids that coexist and don't mix. And this, as simple of an idea as it is really has been paradigm shifting towards understanding basically many, if not all, functions of the cell and how the cell is organized. And so with that, I will hand it over to Steph to start off our first vignette, which is describing the early days and some early history of the field.
All right, thank you. Thanks, Shani. So I started thinking about cell biology in graduate school, and I studied bacteria. And so these bacteria are teeny tiny, like they're microscopic. You can't see them in, with your naked eye. And so what's amazing about these bacteria is they have very little intracellular organization. So this is an electron micrograph of an E. coli cell, and you can see, some dark black like circle spots. Those are ribosomes, and then this white kind of patchy stuff is the DNA inside these cells. And so when I was in graduate school, I tracked the motion of DNA inside the cell. And so what I was really interested in is how do things move inside the cell? How do molecules move inside the cell? And what you'll notice in this movie is that these two DNA segments kind of jiggle back and forth, but they don't really explore the whole space in the cell. They just kind of like jiggle back and forth in place. And so that was a whole, bunch of interesting questions about polymer physics and the DNA packing inside the cell. But the real story that I wanted to get to today, so I moved from bacteria literally up the food chain, and was interested in the C. elegans, which is the roundworm. And this organism is about 1 millimeter long. So you can see adults with your naked eye. They're like a little, teeny tiny comma size. And these beautiful organisms are transparent. And so they're really good for cell biology because we can use microscopy and literally like look through inside the cell. And so you can see here the mouth of the worm. This is the intestine that goes the length of the worm. You can see the gonad here. And in the gonad, there are rows of embryos. And the thing that really got me interested in this field was this movie by Cliff Branglin, our postdoc advisor, who zoomed in on one of these embryos and took a fluorescence movie of this embryo. And what you can see here is there's a protein that is bright white, so it's fluorescing. And you can see dozens, hundreds of small little bright spots, right? And those bright spots start out dispersed throughout the entire cell. And then the movie's looping now. So, and then they disappear in the anterior, in the front, on the left-hand side, and then they appear and get darker and brighter on the right side and the posterior end. And so this was a really interesting phenomenon in terms of how does this cell, which divides asymmetrically, so the anterior cell becomes a different cell fate than the posterior cell, but how do these things that are initially uniformly distributed get inherited by just one cell and not the other? And so I saw this movie when I was finishing up graduate school, and Cliff took this movie in 2009. And it was mesmerizing because these things move very differently than the things that I was looking at inside E. coli cells. And it wasn't at all clear why they disappeared in the anterior, why they reappeared and stabilized in the posterior. And there was just like a lot of like, what is going on? I've shown you a couple different examples of cells so far, right? I've shown you teeny tiny bacteria. I've shown you huge embryos of worms. The cell is the basic unit of life. So I'm made up of a cell. All of you guys watching at home are made up of cells. Plants are made up of cells. Worms are made up of cells. So cells make up every living thing. And so we can sort of reduce life down to a single cell. And so cell biology is the study of cells, right? And how do they function? How do they work? And what I want to emphasize is that like what we're studying in the worm is very relevant to humans and to all forms of life. And so what I was really interested in was how the cell is organized. So Robert Hooke back in the 1600s looked at slices of cork under really primitive microscopes and noticed that they reminded him of rooms in a monastery. And so he coined cells, which is Latin for small room. And so before we dive back into biology, I just want to take a little anecdote into your childhood of the Berenstain Bears. So there's a particular book on the Berenstain Bears and the messy room. And so I want to make this analogy between individual cells and a room, a child's bedroom. And the idea is that, like bedrooms, cells function best when they're organized. So in a child's bedroom, when there's toys strewn across the floor, and brother bear and sister bear are arguing over each other, and there's dirty dishes over here, maybe that creates a health hazard. But the room is very dirty, and things are not going to go well. And so Mama Bear is very unhappy here, and she says, clean your room. And so in a well organized room, toys are in the toy box or arranged on the bulletin board, and everyone is happy and smiling, and there's no safety hazards. And so for a cell to function, the cell has to be able to put all of its molecules in the appropriate place so that it can function, and so that things that aren't supposed to interact don't interact, and things that are supposed to stay together stay together. And so that is the basic question that I have in biology, is how do cells organize molecules such that they can function properly? And so we can think about cells just like the human body, right? So you have a heart and a liver and a stomach, and each of these organs are specialized for a particular function. And the cell is the same way. So this is just like a cut through section of a cell, and the cell contains many organelles, which are just small organs. And each of these have a very specialized function. And not only a specialized function, but they have a membrane surrounding them that is separating them from the rest of the cell. And so the very classic thing that I learned in textbooks growing up, and many of you probably learned this, it's been sort of the dogma for 200 years, is that organelles inside cells are defined by membranes, and they're surrounded by a membrane. And these membranes create a diffusion barrier between the inside of an organelle and the outside of an organelle. And so this black line, this membrane that surrounds the nucleus in this case, allows the nucleus inside to have a different biochemical composition from the surrounding medium. And so the blue molecules stay inside the nucleus and the red molecules stay inside the cytoplasm. But if we look at these other locations in the cell where there are no membranes, then because there's no membrane separating these two compartments, all of these molecules can diffuse around and mix, right? And this is sort of like the messy room, and this is the clean room, right? And so if we have this messy room in the cytoplasm, this is a problem. We want the blue toys to be in the toy box, and the red toys to be over here. And so the cell wants to be able to separate these, and yet without a membrane, it was thought until about 10 years ago that couldn't happen. And so if we go back to the movie of the C. elegans embryo, what was so amazing to me is that all of these sort of spherical spots of bright protein don't have a membrane around them. So how on earth do these proteins stay together in these drops and not just diffuse and disperse throughout the entire cell? And so that was the question that got me into this field, and that was the question that is sort of still driving me. And I'm gonna let Shani take it away from there.
That was amazing. Thank you, Steph. That was such a good introduction to the field. And it was really, it's a good segue, I think, to sort of part 2 of the early days, or how we arrived in this field. The good news is I arrived around the same time, so I was equally interested in a lot of these things, which Steph just introduced so beautifully. But there are some differences, and so I'm just going to point those out here because it's really interesting to see how sort of people arrive from with different backgrounds into the same places and then go off likewise to different areas. So super briefly, my background, initially actually was in physics at CUNY, Hunter College, and then biophysics, biochemistry for my PhD. And my PhD work was actually motivated by understanding a totally different question, right? So it was trying to understand the origins of neurodegenerative diseases. So here, pictured here, is now the sort of famous first Alzheimer, known to be the first Alzheimer's patient, Augusta Dieter. You know, she was led to the first brain autopsy where Dr. Alzheimer here published in this first very seminal paper in 1911, describing the plaques and the tangles in this sort of first Alzheimer's brain. And even the images in the paper are these sort of hand-drawn images of neurons, literally like just describing what he saw in the brain. So sort of these little clumps in and around the neurons and sort of degeneration of the neurons. And so this is a fascinating area, of course, and it's, you know, over a century of research has now been dedicated to understand these origins. So my PhD, like many, many PhDs before me, was focused on this type of question because this phenomenon is not actually even unique to Alzheimer's, although that's the most sort of notorious of the neurodegenerative diseases. This concept of protein aggregation, what we call it, in or around the neurons in the nervous system is actually a feature of so many diseases, including ALS, including Pick's disease, frontotemporal dementia, traumatic brain injury, et cetera. So the focus of my PhD was actually to try to understand how certain proteins result in these fibers that lead to the plaques and the tangles. And the one thing I just want to point out here is that for all of these diseases, while the proteins are different, so sort of the responsible proteins that are thought to lead to these plaques and tangles, they're all different, but they're actually all of the same class. And this class of protein is called disordered proteins. I'm not going to spend time describing what this is, but it's basically it's a class of protein that is very unique in that it's very sort of flimsy and floppy, and it's not sort of a very rigid structure. And so for me, when I was looking for a postdoc, I mean, these papers started coming out. So here on the left is actually a picture of the very same P granules actually in the C. elegans embryo, but here they're in the adult gonad, so they look a little different. And this was back in 2009, one of the first pieces of evidence that demonstrated the liquid-like nature of these P granules, of these germ granules. And another example here in 2011 is the nucleolus, which I think Steph's going to talk about a little bit more in the next section. And so for me, it was very simple. I was used to thinking about, you know, proteins or biomolecules that ended up in this very sort of diseased, aggregated state, this irreversible assembly that was very disease associated, and then was fascinated to think about, well, what do you mean you have these dynamic assemblies, these molecules that come together in these functional ways and give rise to these liquid properties. And so from sort of even a biochemistry perspective, I was really interested in learning how sort of the contrast. So in a way, I was kind of thought I was leaving the field of protein aggregation actually, and doing something a little more fun and dynamic and less disease oriented. But I'll save the punchline, which is that I actually do still study neurodegeneration. And so what I ended up doing in terms of the approach that, you know, my work took me, at least during my postdoc, was to say, so in the state of the field, so around 2012, when I started my postdoc, we knew of these few examples where we saw these, you know, these granules to look liquid-like, is what the literature had called them. So it looks like they didn't have membranes. They looked like oil droplets in water. And that was sort of the analogy. But the thing is, the analogy is just an analogy. So when it comes to oil and water and the fact that these are two liquid phases that can coexist, we understand that. We understand that water is, you know, consists of H2O molecules, and that oils, depending on the oils, consists of these carbon chain type of molecules. And we understand that we understand the differences in those molecules and why they can form different phases and why those phases would coexist and they wouldn't mix together. But when it comes to these complex cellular granules, we have no idea, we knew nothing, right? So we just knew, sort of looking top down, that these fluorescent blobs looked like liquids, and they were composed of thousands of different types of molecules. And so the real interesting question for me coming into this field was, well, what are the molecules? What are the biomolecules that are responsible for this liquid behavior? And what what type of liquid really is this? I mean, are these actually liquids? What type of liquids are they? And can we understand the nature of these liquids on the molecular level, which is again sort of more closely tied to my background. And so I'm going to leave that as a cliffhanger until we get to the next set of vignettes. Okay, so that's what brought us to the field. And now we're going to do our aha moments, and I'll give it back to Steph.
Okay.
Okay.
So I was really interested. I'm going back to the embryo in the worm, and I was really interested in. So the embryo divide. It's a single cell. Starts out as a single cell, right? And then it divides within a fixed volume. And so the cell, divides in half. Shana, can you start the movie?
Sorry.
Sorry. So the cell divides in half, then divides in half again, then it divides in half again, et cetera, right? And so each time it divides, the cell gets smaller. And what I'm showing you here is that in red is the membrane. So you can see the outside of the cell, the boundaries of the cell. And then in green is the nucleus. So the nucleus is one of these membrane-bound organelles. And what I hope you can see is that as the embryo divides and the cells get smaller, the nuclei also get smaller. And so this was an example of this phenomenon called organelle size scaling. And so if you measure the size of a cell and you measure the size of the organelle, then they change proportionally. So even in yeast, so this is true in developing embryos, but even in yeast, the size of the nucleus is really big in large cells and really small in small cells, and you get this beautiful linear relationship, this scaling law between the volume of the nucleus and the volume of the cell. And so so that had been described very well for the nucleus and some other organelles, but I was really interested in this membrane-less organelles, right? And so I'm going to go back and look at the nucleolus. So the nucleolus is one of these membraneless organelles. And here, again, I'm showing you the C. elegans embryo, and it's dividing. And the nucleoli are assembling and disassembling with each cell cycle. And what I hope you can see is that as the embryo divides, the size or the brightness of these organelles get smaller and smaller. And so I was able to measure the size of these using the fluorescence intensity, so the brightness, how white they are. And measure the cell volume using another marker, the red marker in the previous video. And so what I found was that as the embryo divides, so within a fixed volume, these at the 8-cell stage, the cells are large and the nucleoli are large. And as the embryo divides, the cells get smaller and the nucleoli get smaller and smaller, right? And so we have this very robust scaling or proportional change in size between the organelle and the size of the cell. And so I wanted to sort of understand what the functional consequences of this were for the embryo. So the nucleolus makes ribosomes, which are molecular machines that cells need to grow. And so I was really interested in this coupling between size and growth. And so in the embryo, they're obviously not growing, they're dividing within a fixed volume, but it was a nice kind of test tube within which I could test some hypotheses. And so what I did first was I used this genetic perturbation called RNA interference, which is just a way to knock down or silence the expression of particular genes. And so here I have a control embryo, and you can see that it is about 50 microns in length. And then if I knock down certain genes, I could make the embryos really big, or I could make the embryos really small, but I was able to manipulate the size of the entire embryo genetically. And so then what I wanted to do was look at the organelles inside these embryos of different size. And my expectation, based on everything that had been known before, is that the small embryos should have small organelles and big embryos should have big organelles, right? So if we look at my results, boom, totally opposite. So the really small embryos had massive nucleoli, and the really big embryos that actually, you can't even see the whole thing, it was too big to fit in the field of view, but it had really, really tiny nucleoli. And so this was the exact opposite of what I expected. And I had no idea what was going on, and I had no idea how to explain this. And this was me for probably about 2 years of my postdoc, trying to understand what this meant. Because I had seen scaling, right? During development, I'd seen scaling. And so there were these two sort of contradictory results. During development, the nucleolus decreases with cell size as the cells divide and get smaller. But across RNAi conditions, when I genetically manipulate the size of the cell, then I got inverse scaling. So big cells had small nucleoli and small cells had big nucleoli. And this just like did not make any sense. And I didn't know how to sort of reconcile these two results for years. And so finally, I started to think about, okay, how do the embryos get these nucleoli? And so there were two possibilities. So C. elegans, again, you can see here they have this U-shaped gonad. And so what I'm showing you down here is a schematic diagram of this gonad. And so the green spots are proteins. So the proteins that were fluorescently labeled before. And the gonad is what is called a syncytium. So it is open. And so these cells can share cytoplasm with cells over here. And so it's wide open. And what that means is that if the nucleoli are loaded from the hermaphrodite, from the adult worm into the embryo while they're dispersed, then small embryos should have the same concentration of components as large embryos. But on the other hand, if the nucleolus is loaded into embryos when they're pre-assembled, then then small oocytes will have a much higher concentration than large oocytes. And so what I had assumed was that there was a fixed concentration. I assumed that small embryos should have the same concentration as large embryos. And if that were the case, then I should see this size scaling. But in fact, what happened is that nucleoli are pre-assembled when they're loaded into the embryo. And so if we look at the next image, this is the gonad. So this is the U-shaped gonad. And you can see in red the membranes between embryos. And over here, this is the syncytium. So these cells share cytoplasm, but this cell right here, this oocyte, is the first one where the membrane is completely closed. And what you'll see is that the nucleolus is already assembled. And so depending on the width of these oocytes or the size of the oocytes, then every single oocyte receives the same amount of stuff that's pre-assembled from mom. And so that results in a difference in concentration in the embryos. And so what I was able to measure then in the embryos is the concentration of this protein that makes up the nucleolus. And so we could look at the concentration in the nucleus as a function of the embryo volume. And in small embryos, this IMA3 RNAi condition, it's really high. And in large embryos, the C27D9, the names don't mean anything, don't worry about it. I don't even know what this one means, is really low. And so here in the dotted line is the prediction that we would expect if concentration were fixed through just constant and independent of embryo volume. But in fact, it's a decreasing function. And it is related to, so the concentration is just equal to the number of molecules that are loaded into the embryo divided by the volume of the embryo. And since the number of molecules loaded is the same in every embryo, regardless of the embryo size, because it's loaded pre-assembled, then this gives rise to a difference in concentration as a function of embryo volume. And so a consequence of that finally sort of light bulb went off is that this is exactly what we would expect from phase separation. And so if we look at a phase diagram, so this is what is called a phase diagram. So on the x-axis, we have the concentration of this protein. And on the y-axis, we have the state of a cell. And so let's just say we are on this dotted line. And so at really low concentrations, this protein is just dispersed inside the nucleus, and it doesn't condense to form these organelles. But as we increase the concentration above a threshold concentration, this black line here, when the dotted line crosses this black line, then we suddenly start to see organelles assemble. And if we continue increasing the concentration, then the size of the organelle increases. And so that is exactly what I saw when I changed the size of the embryo. And so what sort of resolves this discrepancy that I was so confused about for so long is that phase separation connects not only the organelle size with the volume of the cell, but also with the concentration. And so if we fix the concentration, then organelle size will scale directly with cell volume. But if we change concentration and concentration is inversely proportional to the volume, then the size of the organelle will actually increase with volume because the concentration is, the size of the organelle will decrease with increasing volume because it's the concentration that's increasing. And so that was my big epiphany. And so phase separation was able to resolve this really confusing thing about the scaling of organelle size with cell size. And so now I'll let Shani tell you about her raindrops.
Okay, cool. And I just want to just take a moment and we can discuss it when we get to the Q&A. But I just want to say there's something, I love this story, partially because I was there during those years. And so I think if we even step away from the details of the science for a minute, there's a beautiful aspect of scientific discovery here. And that is, here was the hypothesis. It's going to look beautiful. There's going to be size scaling. And, we'd go to lunch, we'd hang out all the time and be like, it's not scaling. Not only is it not scaling, it's scaling in the opposite direction. What's going on? What's going on? And it was this big mystery and it was crazy. It was the opposite of what we expected. And then I kind of wish the title of the paper is Inverse Size Scaling, blah, 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 sorry. But that was the result, and it was the discovery, was that it wasn't what they expected. And so I actually tell this story all the time when students come to me and saying like, oh no, it's not what we thought. I'm like, that's great, that's okay. Anyway, so I just wanted to add that. So I'm just gonna quickly talk about my aha moment, which I call making it rain. So to remind you, switching gears back, I was interested, it's oil and water, but it's not oil and water, right? What are the molecules that are driving this liquid phase separation in the cell? And this was honestly a very, almost foolishly ambitious project. I couldn't choose between the fishing expedition image versus the finding a needle in the haystack image. So I went with both. We basically said, well, let's just start looking. We know there's hundreds of different molecules in these granules, and we will just make a list, somewhat of an intelligent guess of a list, and then just knock them down one by one, purify them, make them individually, purify, and see whether they will make liquids in a test tube. So sort of what we call a bottom-up approach. So not working in the cell, but just trying to find the purified components and seeing if we can recapitulate these processes. And so in a way, I honestly think I was very naive to even sign up for something like this, but maybe that was a good thing because it's crazy. We didn't know if one molecule was even enough. Maybe you needed 20 molecules of different types. Maybe they had to be arranged in such a way to see this process. So it was really a bit crazy, and it took a little while. And eventually, after looking at a few different proteins, I'm going to gloss over those that 1.5 years of looking for it. We found this one protein that was very, very interesting. It's got the greatest name ever. It's LAF1, which stands for lethal and feminizing, which is a pretty powerful name. And just to cut right to the chase, after all of this looking, we found that this protein, after we purified it, it looked like in the test tube, it turned turbid. So I don't know if you could see, you can't really see that grid line through that first tube, but when the tube warmed up, it got clear again. And so I would run to the microscope as fast as I could to look at this turbid stuff, because it was the one piece of action that I saw after a long time of trying to purify proteins and see if they would phase separate. And I would run to the microscope, and at some point I thought I saw something, but it disappeared very rapidly. And this even has a date and time stamp, which I really like. So this was 2014, March 2014. And I was like, I think I see something, but then it's gone, and nobody even believed me. I couldn't even capture it in the movie initially. And then I saw this, and I'm like, no, there's something going on there. So I fiddled with the buffer conditions even more, and finally was able to make it rain. And so it's fun to think back at this. I've shown this so many times, and now, you know, hundreds of labs have done this with different proteins. The field has progressed so quickly, but at this time in 2014, it was very new. It was very new to the field. It was very new to me. And it was happening at many labs, as most discoveries do, happening at many labs at the same time, similar types of discoveries. And so it was really exciting to be part of that. So here, what you see is that if we lowered the salt in this case, we were able to stabilize these droplets, that they wouldn't disappear like they did before when they warmed on the microscope. And you see them literally raining down onto the cover slip. So these are liquids within liquids. So they're bathed in a larger liquid, and they look like raindrops hitting your windshield. And so this was the moment for me. You know, it was nothing, and then there was something. And then once we had these droplets with this one particular protein, we were able to really dig in and start to characterize these liquids and chop up the protein and figure out all other things that have led to many other discoveries down the way. And so, you know, we could label the protein and measure the fusion timescales of these droplets. And so here you see them properly. We coat the surface now. So instead of wetting onto like they would to your windshield, it's kind of like treating your car with Rain-X. So the droplets sort of bead, and we can look at their fusion and measure their liquid properties. And then we can dissolve the droplets in reverse, so we can disassemble them once, and we can sort of find all the different dials with which we can tune all these different behaviors. And I'm going quickly also, and it's good in time. I think this is a good idea. So that one little raindrop, you know, and then suddenly it went straight to this paper, and we were able to do all of these different characterizations of these droplets. And then this was published in 2015. And so a very cool thing that came out of this work, if you notice the word disordered. So it turns out that this protein LAF1 actually has a domain. So what you do typically in biochemistry is you identify molecule A, and you say, okay, this molecule is necessary and sufficient to do this thing. So then you say, well, what's the smallest part of that molecule that's able to achieve this cool effect? And so this protein had a disordered domain. And so remember how I mentioned disordered proteins are very much implicated in neurodegenerative diseases. And so it turns out us and several labs at the time, in 2015 specifically, we call it the flurry of papers that sort of came out dealing with all of these proteins. Several groups found that the disordered domain of their protein of interest was the necessary and sufficient component to drive the formation of liquids. And so I'm not really going to go into it, but this has partially led to this new emerging hypothesis that actually the misregulation of liquid phase separation of disordered proteins in the nervous system may actually be an unrealized an unrealized pathway towards aggregation that, you know, a century of research has maybe missed, right? Because this is a brand new phenomenon. And so this is only one of the many, many ways that this new paradigm, this new revolution of phase separation is starting to influence various concepts of therapeutics in diseases, as well as understanding the different functions in the cell. And so that's all I wanted to say for that. And I think given the timing, we could break here before we go into pressing questions. I haven't been paying attention to the chat.
Yeah, let me see if I can guide you through some of the questions. First of all, there was a request about how all the things will be disseminated. As you may have noticed, this is being recorded, and so that will be posted. And everyone who registered will also get an email when it's posted. And I think we can also link to, so then you can recover the slides, and we might have a little bit of a reading list or something if people are interested in.
Yeah, that'd be great.
That's pretty easy to do. So there's a set of questions about the scaling of nuclei. So how do you make the nuclei smaller when they've got the same amount of DNA in it? How do things pack?
Yeah, no, that's a great question. So the first, you're absolutely right. All of these cells have the exact same amount of DNA, and so the compaction of the DNA changes as a function of nuclear size. And so there is a lower limit. The nucleus can't get smaller than the DNA, but it's actually so it's not the DNA that determines the size of a nucleus. It's actually a sort of a balance between nuclear import and export of various proteins that form the nuclear membrane, the nuclear envelope. And so actually Rebecca Heald at Berkeley has done some beautiful work characterizing this phenomenon, but it's a totally separate mechanism from how the nucleolus scales with the size of the cell. But yeah, so that is more sort of transport across a membrane to scale the size of the nucleus with the size of the cell.
Good. And then there was the problem of being sure which was the nucleus and which was the nucleolus.
Yeah, sorry, I should have made that clear.
And that's more or less taken care of. There was a question about whether it's harder to purify and identify a disordered protein than an ordered one. And Steph responded in writing with a resounding yes, but Shana, you also
you have more experience.
Yeah. So typically, yes. So especially if a protein is going to like to self-assemble, whether to aggregate or phase separate, then it's actually going to be a big problem. And so one really interesting thing that's happened over the years is so many people have had experiences where they're trying to purify a protein and then the protein just gets lost. I mean, it gets lost for lots of reasons. Protein purification is hard and there's a lot of trial and error, but there are many cases where you're purifying a protein and then maybe your solution gets turbid and maybe you think it aggregated and then you say, oh well, this is a dead end, you move on. I can't tell you how many times I've given a talk and showed those turbid tubes and had people come over to me and say, I was purifying this protein, it has a disordered domain, and my solution's turbid, so I threw them out. And I was like, oh my goodness, just run to the microscope and have a look. It might be phase separating. And so this is one of these areas which it's sort of when we talk about this paradigm shift of field, nobody thought to look for these things, right? So, I mean, a great example, I worked with the protein tau during my PhD. My whole PhD, and again, many PhDs before me have worked with the protein tau. Until people have thought about, the concept of phase separation, we didn't see it. We didn't know that it could phase separate and form liquids. We never saw it, but now we know that it can form liquids, and there's many people looking at whether phase separation of tau is an important early step in tau tangle formation. So it's a little bit of a diversion, but the answer is yes, unless you use denaturing.
And so we've heard about bacteria, we've heard about worms, obliquely, I guess, referring to bigger, more complicated animals. What about plants? Does this happen in plants too?
Yeah, absolutely. So there was a beautiful paper that came out just a couple months ago. So this phenomenon definitely happens in plants. And one of the best examples is a protein that phase separates in response to temperature. And so it actually regulates the seasonal change in protein expression of certain genes, because it phase separates at high temperatures and dissolves at low temperatures, and so that can regulate gene expression in plants. But yeah, there are actually a number of really interesting examples in plants coming out.
Yeah, they're just coming out now, a bunch of papers. There's also phase separation in bird feathers, just as a heads up.
Great. So we have one more quick question, and then that'll leave us with a few minutes for you guys to have last words about where you think the field is going. And this question was whether phase separation has anything to do with the distinction between globular and fibrous proteins.
I'll let you take the fiber one.
So I'm not sure what you mean by globular versus fibrous. Do you mean like soluble versus insoluble, or I don't know if they want to clarify the question.
I mean, one thing that I think Yeah, I mean, one thing I think that is important is, I mean, Shani's right, like disordered proteins certainly are tend to phase separate, but globular proteins can too. So it's
Absolutely.
not only disordered proteins. What actually is most important is the ability to form multiple interactions. So rather than just like binding one-to-one, if you have what is called multivalency, my three fingers can bind like this, or it can bind like this, or like this. And so there are multiple sort of permutations and combinations for proteins to bind. And that is true for disordered proteins because they're large and floppy and can, you know, transiently interact. But that's also true for some globular proteins. And then fibrous, I would at least interpret that as like sort of like polymerization, like in the cytoskeleton, but I'm not totally sure.
Yeah, I think the distinction she's drawing is between between polymeric things like filament, right? Okay.
So absolutely agree.
And yes, I mean, maybe the simple answer to the last question about phase separation and self-assembly is that self-assembly is a big category, phase separation is a big category, and they certainly overlap. I never like to stop people from asking questions, but I think Shana and Steph wanted to give us a couple of moments of their view of what big questions are open, and I think it would be fun to end there. So let me give the floor back to them.
Okay, great. So perhaps coincidentally, I think the pressing questions we focus on are the ones in our labs, but I will let Steph go first.
Yeah, I think we're both gonna talk about going full circle. So yeah. I started my scientific career looking in bacteria in prokaryotes, so I went into the worm for this discussion of the nucleolus. And what I want to point out is that eukaryotes, all these beautiful cells that have these membrane-bound organelles, they exist on this little twig of the tree of life. And so if we, Shana, can you advance that? Yeah. So there's all the worms, plants, humans, everything that you probably know and love is on this green branch, but the vast majority of the tree of life is prokaryotic. And as I mentioned before, prokaryotes don't have membrane-bound organelles. And so I was particularly interested to understand how do they organize their cells. Okay, so my lab has been looking at several proteins in prokaryotes in E. coli, the same cell that I started looking at and that you guys all have in your gut right now, helping you digest your dinner. And what happens is inside these cells, just like in the worm, proteins can phase separate. And so this provides bacterial cells a mechanism to organize, to clean their room, and to form these compartments, even in the absence of membranes. And so I'm super excited about this discovery. And the real sort of, I would say, elephant in the room open question, not only in bacteria, but also in eukaryotes, is what is the function of these compartments? And so we as a field have seen, are able to look at these droplets and the assembly and disassembly of them, but we don't yet have the tools of. All right, so yes, there is an elephant in the room. That is, what is the function of these membraneless organelles? The hypothesis is that it concentrates proteins, into these condensed phases, and that can accelerate biochemical reactions because the concentration is higher. But the truth is there's actually relatively few examples where that's been definitively shown. And partly it's because it's really difficult to control phase separation inside cells. So that is sort of the next frontier. That's what my lab is working on in both bacteria and in worms. And I think there's a lot of interesting questions because in human cells, a lot of phase separation has been implicated in various diseases like neurodegeneration, which I'm sure Sonny will bring up again, and even cancer and even COVID, because it's involved in everything. But we still don't really understand exactly what these things are doing, and so the function is kind of like the big open question. All right. So, Shani, what are your big pressing questions?
Great. So our big focus, that really the thing that we really like to think about is how do individual molecules, individual biomolecules on the nanoscale, so really, really tiny, give rise to these material properties, these emergent, what we call emergent collective properties. So it's actually not trivial to predict how a certain type of molecule, once it can form a phase, what type of phase it will be, how viscous it is, what the surface tension of it is. And it's that's a real major focus of our lab, is understanding these material properties. So I mean, we're interested in material properties, you know, not only from like a physical chemistry perspective, but from several other perspectives. So, you know, one is, you know, of course, how viscous something is, right? We say these things are liquids, and we have evidence that these liquids can range with major ranges of viscosity. It could be as viscous as honey, as viscous as, you know, maple syrup, or something as viscous as like a peanut butter. And we believe these properties are going to significantly impact the functions of these liquids. So, you know, how much, you know, how the mobility within them, right? So how molecules are organized and sequestered, and how they're able to diffuse within a droplet or within a granule, is going to really depend on these material properties. So we care a lot about that in a variety of different areas, including neurodegeneration, of course. And we're also interested in the possibility of engineering novel materials, bio-inspired materials. So if we can understand, again, from this bottom-up approach, what the, wait, don't play the movie yet. If we can understand the rules that govern how you go from a molecule to a material of a certain property, we can not only maybe intervene with therapeutics for diseases where material properties change, like something that goes from a liquid to a solid, say, but we can also create new materials and modulate them as we desire, which is also a very interesting application, and something that we focus here in collaboration with nanoscience and neuroscience here at the ASRC. And so I was just going to leave you off, because actually the advertisement for this actually happened to have this picture here on the top left. So I just wanted to end with that, to tell you what that's about. And so this is this multi-phase phenomenon. So material properties, things like viscosity and surface tension, you know, we think of them as just, you know, characteristics of basic liquids, but they're really important for driving the functionalities in the cell, as I mentioned. And though they also give rise to what we see in actually the nucleolus specifically and other organelles, this multi-phase behavior, where you can actually have one phase nested within another phase in the larger phase of the cytoplasm, say. And so here's an example of a study that we just did, where we looked at a very reductionist system, where we looked at polylysine-rich aggregates and polyarginine-rich aggregates, rich droplets, not aggregates, sorry, droplets. And these are two amino acids that are prevalent in phase-separating proteins. And they're interesting because they actually both have the same net charge. They're both positively charged, but they're different. They're different in their ability to interact, and they interact differently with themselves and with other partner molecules. And so we actually found in this study that the droplets were so different. So the one was more like syrup and one was more like peanut butter, say, in terms of viscosity. And they were so different that they wouldn't mix. So they would form these multiple phases. And then we'll just end with the movie here. So they're so different that if we form one type of droplet and then we add the other component, we can actually see that it competes. Yeah, so here we have droplet of green type, and then we add component of polyarginine, and we see that it displaces them in this very interesting way. And so we could start to build these design rules from these individual components. And I think that's something, just to close off, this is another interesting maybe way to contrast some of the things that like Steph and I are doing, where maybe this sort of like top down versus bottom up approach, both getting at the same, both, you know, coming from the same interest in understanding phase separation, but through these different ways. And that's it. Then maybe we can open it up to discussions. Or I don't, actually, we're a little low on time, so I don't know.
Yeah, so we've had a number of questions. If there are, there's a lot of discussion about microtubules, so maybe we can sort this out. So there was questions about whether the polymerization of microtubule, what's the relationship between the polymerization of microtubules and phase separation, which you sort of answered in the chat. But then there's also the issue of what's going on with droplets when the nuclei are separating during cell division.
So yeah, so I guess I want to be a little bit specific about droplet formation. So so when the membrane-bound nucleus breaks down in mitosis, and then the mitotic spindle, which are made up of microtubules, separate the DNA. And so for some membraneless organelles, they just sort of are segregated by random diffusion because there's enough of them. And some of them dissolve during mitosis, like the nucleoli that I showed you, and then just reassemble in the next daughter cell. And so the sort of the inheritance of membraneless organelles is different depending on the particular type. But then I think there was another question of like microtubules can also serve as a surface for other proteins to nucleate and form other droplets, not nucleoli, but other types of phase separated things on microtubules themselves. So they can actually form a surface to nucleate. So I think tau is one of them, right?
Yeah. So there's some interesting work by the Hyman group. Well, there's a few pieces. I mean, so one is that you can actually nucleate microtubules within a droplet of tau, and analogous work has been shown for actin with actin binding proteins as well, several years before that. And then actually some more recent work from the Hyman group is now talking about this idea of sort of, this wetting phenomenon that occurs on these surfaces specifically. And it's actually really interesting. This is sort of another turn I think the field is taking. So I think initially it was really all about membraneless organelles, right? So it was just like, nothing to do with lipids. These are just, proteins, nucleic acids that are forming these things. And we don't want to think of this in the context of lipids at all. But as the field is sort of maturing, I guess I would say, we're now realizing that there's so many different flavors and versions of this phase separation, some that even involve co-assembling with lipids and co-assembling with membranes even, but there's still being liquid behavior, and then, interactions with other surfaces like, cytoskeletal elements, et cetera. So yeah, it's an interesting question. And I think, the field is sort of evolving to incorporate that now further into the concept of phase separation.
Okay, so we have one last question, which is whether you can envision this going as far as influencing cancer treatments. Sort of,
yes, there was a paper in Science this summer or a couple months ago. I don't know, COVID time warp, but where if you treat cells with various chemotherapeutic drugs, rather than just entering the cell and going everywhere, some of these therapeutics actually, like drugs, partition into and concentrate within some membraneless organelles, and so that has really important implications for sort of dosing and even sort of rational design of drugs to target them to different locations. And so there are a lot of droplets in the nucleus, and so using that to sort of control which genes are or where DNA damage occurs, or which genes are sort of regulated by these drugs is a open possibility, but still a sort of new frontier, I think.
Yeah, and I'll just add to that. So yeah, there's definitely there's several works looking at not only phase separation being involved with the etiology of cancers and the mechanism of it, but then also the treatment, as you mentioned with the Science paper. And so absolutely, yeah. It's involved with everything. I mean, that's sort of the joke in the field. Of course, the answer is phase separation somehow or another, and it seems to be true.
Good. So thank you both. and thanks to our audience. Something I didn't say at the beginning is that we organize these public events many times in conjunction with events that are for professional scientific audiences. And indeed, tomorrow we'll have a symposium devoted to a variety of issues that overlap the problems that you've been hearing about tonight, which actually Steph will be leading us off, which we also appreciate. And we will be doing this whole thing again almost exactly 1 month from now, except the topic will be something completely different, but also something you all may enjoy. So I encourage you to stay tuned. I don't know exactly how you found out about this, but presumably the same path will lead you to what happens next month. We'll have a symposium on problems related to learning both in real biological systems and in artificial neural networks, which are revolutionizing many aspects of technology. And the evening lecture beforehand will be by Jenny Safran from the University of Wisconsin, who's done spectacular work on studying language learning in infants, really pioneered our understanding of the role of statistical structure in language learning. And we look forward to seeing all of you again. So thank you, and thanks to Steph and Shani.
Thanks, everyone.